In this series of forensic science lecture, today we are discussing about the human genetics. When we talk about human genetics, we study various principles related to that. One of the major principles which comes in the human genetic is heredity. What is heredity? Heredity is the transfer of characteristics from one parent to offsprings through their genes. Genes are those substances which are present on our chromosomes, and these are being transferred from one generation to another. When we talk about heredity, we consider that it consists of the parents. One is the father having a sex chromosome XY, another is mother having chromosome XX. Supposedly the father having condition with hemophilia and mother is a carrier. So depending upon which traits or which alleles are being passed to the next generation, will this disease be carried to that particular children or not? In this example, if we just go by the color, we see that the dark represents that the father is having the disease. The mother is not having the disease, but mother is a carrier. Carrier means that the mother will have half the trait of the disease and half the trait which is not diseased. So you can see they have 4 children or 4 offsprings, in which the son having XY, where the X came from the mother and the Y came from the father, and the person, this boy, it does not have any disease. Whereas one of the daughter is a carrier having half the disease trait and half the non-disease trait. Then the another son and daughter, they both are diseased, having the traits come from the mother and the father, which represents the disease. So this is one of the example which I am making you understand through the example of a disease. Same thing happens if we are talking about the phenotypes in the cases of forensic studies. So the phenotype, for example, will look for the skin color, the hair color, the eye shape, the nose shape, the height of the person, how the person appears. So all this depends which traits are being passed from father to the children and from the mother to the children. Depending upon the nature of the traits, whether they are dominant or recessive, the children will appear the same, the genotype which they have taken from their parents. So this is heredity. Then the principles of heredity. Heredity traits are determined by specific genes that specify a certain single characteristic. For example, height, weight, eye color, skin color, etc. Individual, they always carry 2 genes, or we can say 2 alleles for each trait. One from the mother's egg and one from the father's sperm. So depending upon what is being transferred to them, they will look same. When an individual reproduces, the 2 alleles split up and end up in separate gametes by meiosis. Whereas there might be certain differences because of mutation. Now what is mutation? It is the alteration of DNA sequence, whether it be in a small way by the alteration of a single base pair of genetic material, or it be a gross event such as the gain or loss by an entire chromosome. Mutation is a separate topic where we study different types of mutations, what are the alterations which are caused in the DNA, and whether the mutation is significant or not. If the mutation occurs at a specific site and that is being transferred to the offspring, then they will have the mutated DNA with them, and this is how these mutations are incorporated in a population. So to sum up, I will say about the mutation that in one of the parent, if there is any mutation which is being transferred to the son, and then if that is advantageous to that person, then it will remain in the population, and with the time passes, that advantageous mutation become dominant in that particular population. That will look in the Hardy-Weinberg equilibrium. Mutations generally, when we talk about, they are very rare because DNA is a very stable molecule. We know the structure of DNA, which is double stranded. It is made up of two complementary strands, and the repairs can be made by when one strand or other is damaged. But if the both sides are damaged, then the repairs cannot be made in DNA. All the potentially reactive sides group of the sugar molecules making up the backbone of each DNA strand are already covalently bonded. So, when we talk about DNA, we talk about pyridines and purines. We have 4 bases A, G, T, C, adenine, guanine, thymine, cytosine, and how this A always bonds with T and G always bonds with C. A bonds double bond with T and G triple bonds with C. That is how our DNA structure of a stable molecule is being made. So, this bases which we are talking about, A, G, T, C, adenine, guanine, thymine, cytosine, they lie protected inside this sugar phosphate backbone, and their potentially reactive sides group harms were secured by hydrogen bonds. So, they are bonded together, and this DNA helix and nucleosome coils, they provides a geometric tightness which deters most chemical reactions from taking place at normal temperatures. So, to say that when mutation occurs, they are very rare occurrence. Normally, usually when a mutation is occurring, it is because for the benefit of that particular population, that certain advantage is being provided to that population, so mutation is occurring in that particular population. Although when we talk about the evolution, that is one thing, that various mutation comes in the DNA and further which leads to evolution. We say that DNA is resistant to change, but change is must. Mutation helps us to create variations in the gene pool. So, what happens with the natural reaction? Natural selection removes the less favorable from the gene pool, whereas the beneficial or advantageous one tends to accumulate, which results in the evolutionary change. So, we say discuss about any disease. If there is any disease which is occurring in a population, so sometimes because of the natural selection, the poor trait it deletes out, and the new traits which is there which come, and later on it is adapted by that particular population. So, we say the mutation creates variation, and further this mutation we call as evolutionary changes. Supposedly we have a DNA strands, there could be various kind of mutations which can occur. If a particular strands or if the particular bases they gets deleted, then we can call them as a deleted mutation. That will result in less number of bases. There could be inserted, that means more bases came and get attached to a certain part of DNA. Insertions always increase the size of DNA. Another one is substitution. So, what happens in substitution? That a particular sequence is substituted by the similar number of the base pairs. So, in substituted DNA, there could be no change in DNA. Deletion will decrease the number, insertion will increase it, whereas substitution will make the number similar. There will be no change in that. So, where is the mutation occurring? Whether it is occurring in somatic cells or it is occurring in germ cells. The major mutations, if they are occurring in the germ line, those are being transferred to another generation. If the mutations they are occurring in the somatic cells, so these cells they are consequently most mutations are happening which are normal body cells, but they are not affecting the offsprings, at least not directly they are affecting the offsprings. So, but if a mutation is occurring in the germ line, that is being transferred directly to the next generation. So, germline mutations are mostly which have chances to get transferred to the next generation, and they have a chance of being inherited. So, if mutation occurs in gametes or sperms or germline mutations, they have higher chances of being inherited. So, if we take an example, like some plants sometimes can transmit somatic mutations to their descendants asexually or sexually. For example, when flower buds develop in somatically mutated part of plants, so a new mutation that was not inherited from either parent is called a de novo mutation. So, here we have to understand this. If the mutation is coming from any of the parent, we can call it as somatic or germline mutation, but if any offspring develops a new mutation of its own, we call it as de novo mutation. So, this sequence of gene, if we call, can be altered in several ways. Like we have seen, there could be mutations maybe in various ways. It's just not the substitution of the bases, it could be the effect on the structure, which is classified into two parts: small scale mutation and large scale mutations. The small scale mutations, they affect a small genes, one or a few nucleotides, whereas the large scale mutations, they happens in the chromosomal structure. If there is any effect on the function because of the mutations, we call it as loss of function mutations, but if any function is being gained, we call it as the gain of function mutations. So, depending upon what effect mutation is causing on the offspring, we can call as by the aspect of phenotype affected. We can say that this mutation has resulted a specific eye color in this population. So, that is by the aspect of phenotype, कि certain mutation occurred and now this population also having a blue color eye because of this mutation which has happened in DNA. If it is affecting the fitness of the person, that means clinically, medically, it could be that this particular mutation results in any harmful effect, we call it as harmful mutation. But sometimes there are certain diseases which are prevailing in a particular population and this mutation removes that trait, so we call that as beneficial mutation. By inheritance, if it is inherited from any of the parent, it is called by mutation by inheritance, and if the impact of the mutation is on any protein sequence, so we know that there is protein is further coded by the RNAs, and if there is certain impact, there could be frame shift mutations, that means a particular frame is shifted, nonsense mutations, missense mutations, neutral mutations and silent mutations. So, when we talk about if the mutations are causing any effect on the structure, we call them as the small scale mutations. Small scale, as the name suggests, these are the point mutations. So, these are often caused by chemicals or malfunctioning of DNA replication, where exchange of a single nucleotide happens for another nucleotide. Point says that a single nucleotide will be affected. For example, if we have the base A, instead of that the mutation could occur and a person can have another base T on that particular point. So, most common we see in the point mutation is a purine for a purine, that means adenine might change to guanine or guanine might change to adenine, or maybe a pyrimidine for a pyrimidine, that cytosine might change to thymine or thymine might change to cytosine. So, this kind of mutation is occurring, the point mutation when it is occurring, A to G, G to A, C to T or T to C. So, this transition can be caused by nitrous acid, base mispairing or any mutagenic base analogues. For example, one of the example is BRDU, which is 5-bromo-2-deoxyuridine. Sometimes when we talk about that certain chemicals, they are harmful for us, which might cause mutations, which might cause certain sequence change in our DNA. So, we should be taking precautions while using such chemicals in the lab. So, when we talking about the small scale or the point mutation, less common is a transversion, which exchange a purine for a pyrimidine or a pyrimidine for a purine. So, this is point mutations. Changing from a purine to purine is very common, but if a purine changes to pyrimidine or a pyrimidine changes to a purine, that is a less common transition. An example of a transmission is adenine being converted to cytosine. Although we say that this is a less common point mutation, but it might occur in various populations. So, the point mutations that occur within the protein coding regions. Now, we are talking about the regions which codes for protein in our body. So, these are the sequence of the amino acids. So, if there is any mutation changes, that might lead to a certain significant change in the body. So, to understand that, we must know that it is a sequence of 3 bases which we call as amino acid, which further codes for a protein. So, if a point mutation changes that at a specific site, that might lead to the change in the protein. So, it could happen in a way where, depending upon the error in the codon of the genetic code, if that change might lead to nothing, then it is called as silent mutation, that it codes for the same amino acid. If the mutation changes the code to a different amino acid, we call it as missense mutation. But if in certain cases that mutation codes for a stop codon, we have different stop codons which codes for different stopping of that particular protein. So, if it codes for a stop codon, then the protein synthesis will be stopped, which we call as a nonsense mutation, depending upon what changes is being caused by those mutations. It could be silent, missense or nonsense mutations. As we have seen previously in the diagram, there could be deletions, there could be insertions and there could be substitutions. Insertions, as the name suggests, is the addition of one or more extra nucleotides in the DNA. So, whenever any extra nucleotide is being added, it is caused by the transposable elements or maybe due to errors during replication of repeating elements. For example, there is a class of 80 repeats. So, sometimes when the repeat is happening in the DNA, the 80 repeats could be more. If by any chance 1 repeat or 2 repeats are added, then the 4 bases will increase over there. So, these insertions, they sometimes can be reverted by the excision of the transposable element. So, if any insertion happens in the coding region of a gene, it may alter the splicing of the mRNA. That means the site where the mutation happens was the splicing site, or it may cause a shift in the reading frame. For example, if the protein is being read in the terms of 3 and any 80 or any code comes in between, there could be a shift. So, we call them as the frame shift mutation. This both mutations can significantly alter the gene product. However, another thing when we talk about is the deletions. Deletions is any particular nucleotide, maybe one, more than one, maybe AAT, GC, or the combination of AATT, GC, depending upon 1, 2, 3, 4 nucleotides, they are removed from the DNA. So, it all depends from which site the nucleotide is being removed. Like insertions, these mutations can alter the reading frame of the gene, but we must say when we talk about the insertions, they could be reverted, but if any deletions can happen, they are irreversible in nature. This we were talking about the small scale mutations which are happening. When we talk about the large scale mutations, they are the amplifications or gene duplications which leads to multiple copies of all the chromosomal region, which increase the dosage of genes located within them. Or maybe amplification is one part. Large scale also depends the deletion of large chromosomal regions leading to the loss of the gene within those regions, and chromosomal translocations which interchange the genetic parts from non-homologous chromosomes. So, this chromosomal translocations could be there, deletions could be there, or amplifications could be there when we are talking about the large scale mutations. So, in this, an intrachromosomal deletion removes a segment of DNA from a single chromosome, that is known as interstitial deletions. A chromosomal inversion is reversing the orientation of a chromosomal segment. Whenever we have a chromosomal inversion, whatever chromosomal segment is there, it could be inverted. That means the part which was at the top, it comes to the bottom, and the part which was at the bottom, it goes to the top. It may lead to the loss of heterozygosity. That means loss of one allele either by a deletion or recombination event in an organism that previously had two different alleles. So, this loss of heterozygosity might lead to removal of one allele. For example, we say, just take the same example of the height, that capital T and small t were two alleles present in a population, where capital T determines the taller height and small t determines the smaller height. So, because of the loss of heterozygosity, a large scale mutation, a particular allele is being removed. Now, the population only has the allele of the taller height. That means all the offsprings who will come from this population, they will always have the children who will have a taller height because of this mutation. So, we cannot say that every time the mutation is whether beneficial or harmful, depending upon what kind of effect it is causing. If it is stopping any function, it could be harmful to the individuals. If it is not stopping any function, then we can say it is not beneficial also. But sometimes if any bad gene related to any disease is removed from the population, then we call this mutation is beneficial. So, if we discuss from the basis by the effect on the function, this loss of function mutations are the result of gene products having less or no function. When the allele has a complete loss of function, it is often called an amorphic mutation, and phenotypes which are associated with such mutations, they are often recessive phenotypes. Whereas the gain of function is when the gene product gains a new and abnormal function. These mutations usually have dominant phenotype and we can call them as neomorphic mutation. Dominant negative mutation having an altered gene product that acts antagonistically to the wild type allele. In such cases, it results in an altered molecular function which is often inactive and is characterized by a dominant or a semi-dominant phenotype. There might be in certain cases that a mutation lead to the death of the organism which carry mutation. So, one of the disease which runs sometimes in a family, if it is running in a career mode, then nothing is happening to the offsprings. But there are certain cases that if that mutation comes in the form of homozygote, it might lead to the death of that particular offspring. So, lethal mutations are very harmful, which may lead to the death of the organism which is carrying that particular mutations. There are certain disorders which are going on in the populations and which can cause the lethal mutations or death of the offsprings. There are back or reversion mutations, which is a point mutation that restores the original sequence and original phenotype is hence restored. So, this is by the effect of the function. Going to the aspect of the phenotype affected. If it is affected by the morphological changes, for example, height change or by the skin color change or by the shape of the nose change, then we call is that phenotype affected, or the biochemical mutation which results in the stopping of any particular enzymatic pathway. Often morphological mutants are the direct result of a mutation which change due to the enzymatic pathways. So, these two types of mutation result in the effect of the phenotype. Phenotype, as we discussed, which is visible to the other human beings. By the effect on fitness, it could be neutral mutation or the deleterious mutation. Neutral has no harmful or beneficial effect, deleterious which has a negative effect, that means incorporating any disease into that. As we discussed, it could be advantageous mutation or nearly neutral mutations which could be advantageous or slightly harmful. Sometimes we have mutations from inheritance, and as we discussed, if the mutation is causing any impact on the protein coding, then they are termed as the protein sequences mutations, which could be frame shift, nonsense mutation, where could be a premature stop, a missense mutation which is caused by the point mutation, and single mutation could change the substitution of different amino acids, where neutral mutation where this nothing happens, if AAA is also coding for lysine, AGA will also code for lysine. Silent mutation which occurs in a region that does not code for a protein, therefore, it does not result in any change to the amino acid caused. One of the special case is a mutation that has wild type phenotype and under certain permissive environmental condition, and a mutant phenotype under certain restrictive condition. For example, a temperature sensitive mutation can cause cell death at high temperature, but might have no consequences at a lower temperature. So, these mutations, they can be studied in various populations for the population genetic study, and that can further help us in various forensic genetics cases, as well as to identify the individual, which ancestry they belong to, which particular community they belong to, and which particular caste they belong to, which helps us in solving the various forensic cases. Thank you.